I am a kidney doctor, and it's one of those things that, you know, when I was an internal medicine doctor, kidney medicine was kind of some black box that you kind of like, oh, it's a kidney problem, like send it to the kidney doctor. And so most people who are like doctors didn't really learn a lot about kidney medicine when I was growing up. And so what I want to talk about today is something that I've become more interested in over the last couple of years. For many, many years, I've been working with a guy named Tony Sebastian, who just recently died, who was interested in the effects of acid balance in people whose kidneys were relatively healthy. So when you have kidney disease, many things fail, and one of the things that happens is the kidneys are unable to get rid of the acid in the body. And if you look at most kidney textbooks, what they still say is that until the very, very end, your body is in homeostatic balance and the acid isn't doing anything. And so therefore you don't need to worry about it. And Tony didn't believe that, and he started looking at whether or not high acid balances could actually be found in people whose renal function was relatively normal. And so, and from that, and this is data that goes back maybe about 40, 50 years now, we've begun to look at, can you look at acid balance in people whose kidneys have not failed? And this brings me to what I'm going to talk about today, which is how things that happen in kidney failure are very similar to things that happen in aging. And as a kidney doctor, one of the things that we're particularly interested in is, are there any modifiable factors? I mean, there's a lot of things that we can't do anything about, and very little that we can do to try to slow the progression of kidney disease. And so we're always looking for things that we could potentially change. And certainly when I was in medical school, aging was one of these things that we didn't know very much about. And now we know at least two different factors that are associated with aging. And one is what happens to telomeres as you get older, and then the other one is this molecule called klotho. I'm going to talk about those more in detail. So it was fascinating to me that there are a lot of similarities between the effects of uremia and the effects on aging. And and I'm going to say that a lot of this talk is going to be theoretical. So I'm going to talk about what do we see in uremia and how can we modify it in uremia, and then is this going to be translatable to the same effects on aging, because the molecular mechanisms are similar. So I don't know if this is true or not. I think that it's interesting and testable. Okay, so they have things like they both increase TGF-beta, which is involved in fibrosis, goes up. The ability of the cells to get rid of damaged organelles goes down, that's autophagy. You get increased programmed cell death, and you get an increase in the stopping of the cell turnovers. This is associated with telomere shortening, and the shorter your telomeres are, the shorter the period of time that you live. In addition, klotho levels go down as you get older and as you develop renal failure. You get increases in advanced glycation end products. You get increase in mitochondrial dysfunction, and there are a couple of things that may be going wrong with the mitochondria. Some people feel that it's due to increased reactive oxygen species. Some people feel that it's due to increasing damage of the organelles in the mitochondria. And just like inside the cells, where you have decreased pickup of the organelles in the cells, you have decreased pickup of the damaged organelles in the mitochondria. And then what I'm going to talk about is certainly in renal failure, as renal failure progresses, your ability to get rid of the excess acid load goes up, the increases in acid go up, and you develop what we call, you go from just having your pH be relatively low and your bicarb be relatively low, to actual clinical metabolic acidosis. And in aging, the older you are, the less likely you are to be able to get rid of the acids. And that may be related to kidney function. And so we see a bunch of things that happen in uremia that we also see in aging. So it is clear that as kidney failure progresses, you lose bone mass. And it is clear that as kidney failure progresses, you lose muscle mass. And it is of course clear that when you have renal failure, your kidneys aren't working so well. But on top of that, we also see these same things in aging. So once you hit about 30 years old, your bone mass starts to decline. It declines depending on what your peak mass was, but everybody's declines over time. Same thing with muscle mass. And I'm going to talk a little bit about GFR a little bit later on, and that's glomerular filtration rate. But on average, if you look at population studies, on average, kidney function declines with age. So now we'll talk about the potentially modifiable renal factors. So I'm going to talk about kidney function, clotho, and the effects of dietary phosphate. We'll talk a little bit about telomeres, and here I want to talk about stress, because I absolutely agree with Dr. Malhotra about the effects of stress. And then finally, I'm going to talk about some of the things we know about alkali therapy. Okay, so kidney function. So within the normal human pH range, which is 735 to 745, doesn't matter whether you're on the high end or the low end. So when you're born and you have about 2 million nephrons, oops, which one is the light thing? Here we are. Excuse me, I'm going to just screw this up. Okay, so when we're born, when we have about 2 million nephrons, we regulate our blood pH at about 743, 744, with our plasma bicarbonate being around 26, 27. And when we're 80 years old, so 6 decades later, we regulate our blood pH 736, 737 with our bicarb 22, 23. And that's considered to be normal. And I'm going to suggest to you that that's pathophysiologic. So let's talk about acid balance. So acids come from the foods that we eat and from endogenous metabolic processes. And the acids are either neutralized or buffered inside the body. So they're buffered inside the red cells, and they're buffered inside or neutralized inside the muscle cells by breaking down the muscle proteins. They're neutralized inside the bone. Bone is mostly alkali, and so you break your bone down in order to neutralize the acids. The vast majority of acid is turned into carbon dioxide and excreted by the lungs. And all the other acids are excreted by the kidneys. And if we say in foods, what kind of things are we talking about here? And this is just chemistry. Okay? So from the sulfur-containing amino acids, you get methionine or cysteine, you get sulfuric acid, you break glucose down into lactate, triglycerides into acetoacetates, nucleoproteins into urates, phosphates, and then chlorides. Most of the chloride in our diet comes from table salt. And so just to kind of talk, what did I just say? I believe that aging and renal dysfunction have a lot of similar pathophysiologic characteristics, and that with increasing age and with renal dysfunction, the steady state levels of the acid in your blood and in your tissues goes up, okay? And that, and we'll talk about that. And then diets are the source of the vast majority of the acids that we get. And we, meaning if you're eating a Western diet, you're eating a high acid diet. Now let's talk a little bit about what do we know about some of the things that affect how long you live. Well, one of the things is if you put these fuzzy animals on a sulfate-restricted diet, then the animals live longer. And if you put them on a calorie-restricted diet, they'll live longer. And in cats, if you put the cats on a phosphate-restricted diet, then they live longer. So their blood phosphate levels go down, levels of parathyroid hormone go down, and cats who have a tendency to develop renal failure. So the cats on the low phosphate diet live much longer than the cats eating the high phosphate diet. And cats would normally eat a really high phosphate diet because they're carnivores. So what do we know about what happens in human beings? So if you're eating a high phosphate diet, and what's a high phosphate diet? So that's dairy products, legumes, chocolate, nuts, but really phosphate's in everything, because phosphate's an intracellular ion, and so pretty much everything you eat is going to contain phosphate. It's just a question of how much. So here you are, your kidneys are fine, you're eating a high phosphate diet. In order to get rid of that phosphate, what you do is you increase the amount of FGF23. So FGF23 acts as attaches to the FGF receptor, and clotho is a co-receptor for this complex over here. The clotho is then broken off by the secretase, and it floats around until it gets to the proximal tubules. And inside the proximal tubule, what it does is it gets rid of the phosphate the sodium phosphate transporter that allows the body to reabsorb phosphate from the urine. So the clotho comes in, it gets rid of that transporter. That means the phosphorus stays in the urine. You dump the phosphate in the urine, your phosphate level, your phosphate balance goes back to normal. That's what we normally do. So now you keep eating your phosphate diet, and now your kidneys aren't working so well. And so in order to compensate for that, you have to keep raising your FGF23 levels. And FGF23 is a factor in the production of 1-alpha hydroxylase, which turns 25 vitamin D into 125 vitamin D. So the higher the FGF production, the lower the amount of 125. And 125 is a cofactor for the production of clotho. So phosphate goes up, the FGF23 goes up, vitamin D and clotho go down and down and down. And as you get older and as your kidneys function keeps declining, you keep getting lower and lower levels of vitamin D and clotho. So now let's look at the animal models. So if you look at, which is really the only way you can study this, if you look at normal, what are these? Mice. If you look at normal mice, this is what a normal kidney looks at. If you knock out the clotho gene in these mice, what you see, so that means that the mice are just going to keep reabsorbing all of the phosphate that they see. So the kidneys are essentially calcified, and not too surprising, the kidney function is much worse. Or if you knock out the phosphate receptor, but then you put the animals on a low phosphate diet, it doesn't matter too much because they're not eating a lot of phosphate. But knock out those receptors and put them on a high phosphate diet, and now the kidneys are damaged again. And so that's if you knock out the clotho gene. And so if you just look at animals then, and this is a model of kidney failure in these mice, okay? And then you put the mice on a phosphate-restricted diet. So these mice that would develop uremia anyway, okay, have higher clotho levels if you put them on a very low phosphate diet, and this is particularly true as they get older. And if you take the clotho knockout mice, which is this little tiny mouse over here, okay, and you put that mouse on bicarbonate, then that mouse on the bicarbonate with the clotho knockouts live much longer than the clotho-deficient mice not put on bicarbonate. Now, to give you an idea, a normal mouse would be like over here. So these mice die faster. They just don't die as fast by giving them bicarbonate. So if I say to myself, do I think that perhaps, there's a role with clotho and kidney function decline, and, is it possible that this is related to, acid intake? I'm going to say maybe yes, okay? There's some evidence that lowering sulfate and phosphates might help keep, small fuzzy animals alive, or phosphates certainly in cats. And if you give these animals alkali, they live longer. So that's animal data. That doesn't necessarily mean it translates into people data, but it's a beginning. Okay, and now we'll talk about telomeres. So telomeres are TTA GGG repeats, which are at the ends of the chromosomes. And what they do is they tell the DNA repair that those are the ends of the chromosomes and they shouldn't fix that part. And as you get older and older, so when you're young, your chromosomes have long telomeres on the end, and telomeres are added by this enzyme telomerase. So the more telomerase activity you have, the more you keep adding the telomeres to the end. And as the cells get older and older, the telomeres get shorter and shorter, the activity of telomerase goes down and down, until finally when you get to the end of the cell cycle, the chromosomes no longer have telomeres and your telomerase activity is really low. Okay. So these were studies done by a friend of mine, Alyssa Epple at UCSF. And Alyssa worked with Elizabeth Blackburn, who was the woman who won the Nobel Prize for telomeres, who's also at UCSF. And Alyssa did these studies in caretakers who took care of chronically ill people. And she did a couple of different things. So the first thing she did was she looked at telomerase activity, and these are healthy people, and then these are the caregivers. And they stressed them out by making them do timed video games and talk to an audience. And so what you saw is that acutely, telomerase activity went down, and then when they stopped, actually it went up higher than it had been in the first place. But the caregivers had really low telomerase activities to begin with, and it never got nearly as high as in the healthy people. And if you looked at how long these people had been caregivers, you could see that telomere length went down the longer they'd been caregivers. And Alyssa believed that this was related to stress. The way she thought about it was that there had to be biochemical effects that was somehow doing something to inflammation and oxidative stress. And what she found is that the groups that had the highest level of cortisol and the highest level of the catecholamines, norepinephrine and epinephrine, were the ones with the lowest amount of telomerase activity and the ones with the shortest telomeres. So this is a direct biochemical relationship or association between the amount of stress and these specific hormones which are related to stress. And when they looked at it, they could correlate an increase in oxidative stress markers, and I think she was using F2 isoprostane to vitamin E ratios. So higher the oxidative stress, the shorter the telomeres, the lower the telomerase activity, and the more this correlated with the number of years that they'd been caregivers. And when you looked at cardiovascular risk factors, and as a kidney doctor, you don't die of kidney disease, you die of heart disease. What they saw was that their resting heart rates were higher, their heart rate beat-to-beat variability, which is an indication of how flexible, how much reserve does the heart have, Their beat-to-beat variability was lower, their LDL cholesterols were higher, the total cholesterol to HDL was lower, their fasting blood sugars were higher, they were more likely to have metabolic syndrome. So all the bad things that we talk about when we talk about heart disease, actually what Dr. Malhotra was just talking about, all of this was associated with having shorter telomeres and less telomerase activity. And when you look at diet and you look at the effects of telomere length, essentially what you saw was that the more bad foods you ate, so processed foods and junk foods and whatnot, the shorter your telomeres were. And the more likely you were to eat either a Mediterranean diet or have one of these high, forget exactly what this stands for, health equivalency scores. If you have one of these high scores, and this would be like the Ornish diet or maybe the low carb diet, then you were more likely to have longer telomeres. So there appears to be some relationship between what you eat and your telomere lengths. And if oxidative stress is important to this, I kind of wanted to point out that foods that are high in antioxidants are actually often low acid foods too. So what are low acid foods? Well, since I just said that all foods contain acids, but fruits and vegetables in particular contain compounds that are metabolizable to bicarbonate. And so if you're eating a really high fruits and vegetable diet, you're going to be eating a relatively low acid diet, and also potentially like a relatively high antioxidant diet. And the only study that I could find that actually looked at some dietary acid factor was this one from Glasgow, where they showed that the higher the phosphate intake, one, not too surprising, the worse your kidney function was. Also not surprising, the lower your vitamin D levels were, and this would potentially correlate with Klotho levels too. Higher the levels of inflammation were, and the shorter your telomeres were. So, potentially, a link between the acid in the diet and the telomere length. And so just to kind of summarize this part a little bit, it's really clear that telomere length and telomerase activity correlates with stress. There are biochemical reasons to think that this is a relationship, and that potentially, both oxidative and acidative stress may be contributing to shortening telomeres, which are associated with aging. And that, again, makes you ask, is there something that we can do about this? Because I'm interested in modifiable factors. And that brings me to alkali therapy. So just to show to you, just to trying to prove to you that a high fruits and vegetable diet is a low alkali diet. So we did this study where we took all the calories from junk food and whatnot, and we turned it into calories from fruits and vegetables. So essentially 2/3 of the calories in the diet came from fruits and vegetables, compared to, and this is the, compared to the American Association diet, American Diabetes Association diet. And so you can see that if you eat this diet, you are eating a low acid diet compared to a typical American diet. So high fruits and vegetables, low acid. So now let's talk about some of the human studies where they gave alkali therapy and then they looked to see what happened. So in this study done by one of my colleagues, Rado Kraft in Switzerland, he put older, meaning like 60s to 80s year old people on potassium citrate or placebo, and then he looked at their bone density after 2 years. And so in blue is the placebo group, and in red is the group that was given potassium citrate. And so you could see that after 2 years, the group that was on the alkali had better bone mass. And so if you're eating a high acid diet and you're elderly and your kidney function isn't as good, that high acid diet may be breaking down your bones, and if you give exogenous alkali, maybe this will help prevent that because you're taking in the extra alkali. You don't need to break your bones down in order to supply it. In addition, in subjects who have advanced kidney disease, not yet on dialysis therapy, if you gave them sodium bicarbonate, you could improve their nitrogen balance. And this is a big deal in people with advanced kidney disease because they're breaking down their muscles because of the acid in their system. And when you really have advanced kidney disease, you lose your taste for protein. In fact, people will tell you, I don't feel like eating meat anymore. It doesn't taste good. So it's one of the things we look at when we try to decide how uremic you are and do you actually need to start on dialysis yet, okay? And then finally, it helps prevent progression of kidney disease. Now, about 10 years ago now, an Italian group showed in people with advanced kidney disease, so their GFRs were, you know, like if we start people at a GFR of 10 on dialysis, their GFR was like 20 or 25. If you gave them alkali therapy, then you could actually slow the progression of this advanced kidney disease, which is really hard to do, and keep them off dialysis longer, okay? Their kidney decline slowed. And what Donald Wesson showed is that even people who had much better kidney function, and this is called stage 2 kidney disease, but a GFR of 85 is about what I would expect your GFR to be if you're 55 years old, okay? And if it's like, you know, down to 60 over here, that's your GFR when you're about 80 years old. So this would be a typical GFR for somebody who's an older adult. And as you can see, giving these people bicarbonate over many, many years helps slow the decline in kidney function in these people who are otherwise relatively healthy, at least in terms of kidney function. So, and now let's talk about that whole thing about average decline in kidney function with age. So many years ago, there was a study called the Baltimore Longitudinal Study on Aging, which was done by a very famous physiologist called Nathan Schock. And they followed men in Baltimore over about 2 decades. And every couple of years, they did another 24-hour urine collection, and they looked at the rate of decline of kidney function. And it turned out that about a third of the patients didn't have a decline in kidney function over time, and they couldn't understand why that was. So they divided them up into people who were healthy and people who had high blood pressure and people who had other problems. And even so, there was just this group whose kidney function didn't get worse as they get older. And so, which is interesting. And makes you say, well, you know, so what's going on here, okay? So we looked at one of the studies that we had done where we gave potassium bicarbonate to women who were at least 5 years postmenopausal, and we did 24-hour urines like every 3 months. And then we just looked at the slope of the creatinine clearance over time. And so there were a whole bunch of women whose kidney functions didn't change. But these women down here, these were the women with the highest acid load intake. So even in this group, which is, you know, we didn't follow for nearly as long as these people were followed, these people over here were the ones with the highest diet acid load. And these women did not. And now finally, let's talk about this study by Dean Ornish. So Dean actually did this study with Alyssa. And the Ornish diet is this low-fat diet where you also do a lot of exercise and you do a lot of stress reduction, and he's into having what they call love. So you increase your social connectivity. And this was in men who had prostate cancer. And so they did this whole Ornish thing for 3 months. And then they looked at telomerase. And so in the little squares over here, in these guys with prostate cancer, okay, he was able to show that telomerase levels went up as you improved your diet. And so what I want to really try to get across is that if there's a possibility that there's modifiable factors in aging, okay, and if these have anything at all to do with what we see with kidney disease, okay, if there's a relationship between the pathophysiology in kidney disease, which we know more about, and the pathophysiology of aging, which I'm suggesting is similar, okay, then these are potentially modifiable factors, so that potentially we may be able to do something about this, okay? Personally believe that, you know, it's not just one thing, it's, you know, a combination of things like good diet and exercise and lowering stress levels, and that all of these things are important. But if you can actually, you know, I mean, we're all going to get older, but maybe we could get older healthier, and, you know, wouldn't that be a better thing to do? And potentially we can test this. And so that's actually something that we're trying to do now, looking at some of the renal databases. We've been applying for grants to try to measure clotho levels and telomeres so that they have the longitudinal data. We just need to measure the biomarkers so that we can see if there's a relationship. And so maybe this will help you live longer and healthier. And so here's summer in San Francisco. This is what we left. And thank you very much.